I am going to be talking to you today about total protein and protein normalization with our Simple Western systems. So as Courtney mentioned, we've got a whole slew of online workshops coming your way. We've already covered a good bit, but in 2 weeks, we've got Julia talking about Stellar. April 13th, we're going to have Brendan talking about Compass 6.2 and different annotations with that new Compass software. April 27th, we'll have Chia talking about Empower software with Maurice. And then we'll have Benjamin on May 11th with Ella Open Cartridge. We'll have Shihab on May 25th with MFI. And then we'll have Faraz on June 8th talking about Maurice Turbo. And all of our online workshops will be held on Thursdays at 1 p.m. Eastern Time or 10 a.m. Pacific Time. Aside from maybe one or two, but those will be denoted in the online academy. So this presentation is kind of building off of what we've talked about thus far. So here we're assuming that you've already determined your antibody saturation conditions and the linear dynamic range of your sample. So now it's time to normalize your target to eliminate any technical errors and determine any biological differences that you're seeing. So today we'll cover the importance of normalization, types of protein normalization, the total protein assay, and protein normalization on Simple Western systems. So let's go ahead and get started. There it goes. Okay. So let's start off with why normalization is important. So normalization accounts for lane-to-lane variation, allowing for a quantitative comparison. If you're not normalizing your targets, you can't truly determine if the differences you're seeing are biological differences or experimental error. So there are two main types of normalization right now. There is normalization to a housekeeping protein and normalization to total protein. So traditionally, housekeeping genes such as beta-actin, GAPDH, and tubulin, they've all been used for protein normalization in Western blotting because they are highly abundant and they have been thought to be expressed at a constant rate. It has come to light, though, in recent publishing that housekeeping proteins are not quite as consistent as we once thought. Recently, normalization to a total protein quantity has entered the scene as a more accurate way of determining fold changes in your protein expression. So this method quantifies the total protein in a sample in an antibody-independent manner and minimizes the impact of varying expression of a loading control. So each method has its strengths and limitations. There are different validation processes and requirements. So the choice really depends on the context of your experiment. This is a great table to keep in your back pocket when you are considering which method is best for you. Now, a couple things I want to highlight is for each method, you must determine the linear dynamic range to ensure that that range overlaps with your target. For internal reference proteins, housekeeping proteins, you really only have one single point of reference. So it's also important to note that because these are highly expressed, this may limit overlap with your target protein, and you must also ensure that your housekeeping protein is not impacted by experimental conditions. Now, Simple Western offers two different options for total protein normalization. We have the total protein assay and protein normalization. Total protein assay uses a chemiluminescent labeled dye that binds to all primary and secondary amines in the capillary. And you can use this assay with both the big box and the small box instruments. If you're using this in conjunction with an immunoassay, this method will either require replex or it will require you to use separate capillaries from your immunoassay. We also offer the protein normalization kit, which uses a fluorescent dye that binds to all of the primary amines in the capillary. And this method is only available for use in the Just system since this is done in the fluorescent channel. But this kit can be multiplexed with your immunoassay. So let's talk about protein normalization in the Simple Western systems. Simple Western consists of both the big box and the small box instruments. The big box instruments, which are the Peggy Sue and the Sally Sue, can process up to 96 samples and separate based on size with chemiluminescent detection. Our Peggy Sue instrument also offers the option of charge separation. We also have the small box instruments, which are the Wes, the Abby, and the Jess, and these can process up to 24 samples. Now, these instruments separate based on size with chemiluminescent detection, and Jess also offers the option of 2 channels of fluorescent detection. And you can see from this table the options for total protein and protein normalization for these systems. So TPA, labeled right here, is the total protein assay down in the chemiluminescent channel. This is available in all of our instruments, but some of them you will have to use separate capillaries unless you have the option of replex, which is available in both the Jess and the Abby. Protein normalization, as I said before, is only available in the Jess instrument since this is a fluorescent channel protein normalization kit. So now let's talk about normalization using total protein assay in our Simple Western systems. The Simple Western total protein assay uses a biotin label that binds to all primary and secondary amines in the capillary. Then streptavidin and HRP then binds to that biotin label, and detection is done with chemiluminescence. So to the right is a data set of an immunoassay combined with the total protein assay on Wes. You'll notice that different separate capillaries were used for the immunoassay and the total protein. I also want to mention that if you are doing the total protein assay, you'll want to include a key control, which is the no biotin labeling agent. And this is to determine any endogenous biotin species in your sample. And you can do this by replacing the biotinylating agent with antibody diluent, and then continuing with your normal steps of streptavidin and HRP and luminal peroxide. So in instances where you may not be able to use replex, such as Wes or the big box instruments, you will need to put the immunoassay and the total protein assay in separate capillaries. Now, this is a great example of a plate layout that you can use to set up that type of run. And you will choose a run for the total protein size, and then put the target protein in one capillary and the total protein assay in the other capillary. And you'll continue with the total protein assay as you normally would, using blocking buffer and primary, and then the other one will use the label and blocking buffer in place of the primary. You'll notice that there is an extended time for the blocking step for 30 minutes. This is for the biotin labeling time. So now I know that many of you now have a Jess or an Abby instrument, so let's talk a little bit about replex. Replex is our proprietary kit that allows for the removal of your first probe and a subsequent reprobing with a second probe. So this is similar to how a strip and reprobe on traditional Western blotting works, but since our proteins are cross-linked to the capillary wall, our replex process is at least 95% effective based on our data. And I've included an example of a plate layout if you are performing an immunoassay and a subsequent total protein assay on a Jess or an Abby. You will notice that added reagent, the replex assay, has been added to the replex reagent has been added to this assay, and you'll have your first probe as well as your total protein probe in the plate. So with the Compass software, the normalization of your targets will be performed for you. So you will have columns for both the area for your peak as well as the corrected area for your peak. And these are highlighted in the boxes here. You can also visualize the total protein assay in the lane view and look at the probes side by side. So here in the chemiluminescent channel, just under that 40 kilodalton marker, you've got your immunoassay, and then right next to it, you've got your total protein assay in probe number 2. All right, so now let's talk a little bit about our other method, the protein normalization kit. And just a reminder, this is performed in the fluorescent channel, so it's only available for use on the Jess instruments. But for protein normalization, a proprietary fluorescent labeled dye binds to all the primary amines in the capillary. And this assay can be used in multiplex with your immunoassay, so there's no need to do replex for the protein normalization kit. And just like the total protein assay, Compass will do the calculations for you. It will calculate your corrected area automatically. And you can also double check the overlay of your protein normalization by clicking on the blue channel where you've got your different channels up at the top. So at this point, you might be wondering what is right for me. So we've got a workflow for determining that. Probably the biggest factor in choosing your normalization method will be the linear dynamic range of your target and for your normalization method. So these linear dynamic ranges must overlap in order to be used together. So an example workflow to determining the linear dynamic ranges is to perform a lysate titration, and then you will use this to determine your linear dynamic range for both your target and your normalization method. You'll find where those ranges overlapped, and from there, that is the optimal concentration where those ranges overlap, and you can go and optimize your assay from there. So this is a great table made by my colleague, Courtney Garibian, and this shows a sort of workflow for choosing your best normalization method. The lysate concentration, as I said, is probably the most critical factor in choosing your normalization method. So the next question is, are you able to multiplex or do you need to replex? And these factors will differentiate if you can do the total protein assay or protein normalization with your target. And if you're not sure, you can always contact your FAS. We're always happy to walk it through with you. If you're interested in learning more, you can check out our app notes, tech notes, and the user guide for simple Western normalization. You can also contact your local FAS if you have any questions or if you need any help choosing whatever is right for you. And with that, I am happy to take any questions that you guys may have.
Pretty quiet in the chat. Monica, do you have any suggestions of how the reagent might be prepped or how to increase the reproducibility and stability of the reagent or maximize the stability?
For the total protein assay or protein normalization?
Yes.
So these kits are one-time use. You would prep them the day of and then load them into the plate with the rest of your reagents, or any of your other simple Western boxes will take care of the rest for you.
Awesome, thank you.
So Monica, the question that often comes up is that if people have 3 sample sets and they have to do 3 separate runs, can these sample sets be normalized as one set?
Yes, you can normalize them as one set. I would recommend putting in some sort of internal reference control or something like that to account for any plate to plate or instrument to instrument variations, and then normalizing from there.
Sounds good. Thank you.
We've got one in the chat. How does the protein normalization process interact with downstream antibody detection? So I know that the protein normalization reagent is put on before your immunoassay detection is performed. So we have not seen really any interactions that I know of, but I think it's always a good idea to include some sort of control in there, maybe one well without the protein normalization kit with your target, and then look at if there's any differences in that signal.
I'll give it just another second for you here, Monica. I can see someone's typing in the chat.
Phil helped answer that question. Our R&D department tested a variety of targets and antibodies during the development and had little to no effect on the detection of the downstream antibody detection with protein normalization. We've got a couple of other people typing in the chat, so I'll hold. You show total protein and protein normalization at 0.2 mg per mil concentration. Where do you draw the line, i.e. if your lysate concentration is 0.2 mg per mil, what method would you recommend and why? So at that point, I would probably recommend the total protein assay. It's got a little bit of a wider dynamic range. I believe we have used it up to 0.4 in practice, but we do like to keep it under 0.2 ideally, mg per mil. What is the range of concentration used for the lysates? So for that, I'm going to refer back to one of the slides on here. It depends on which one you are using. The total protein assay is from 0.01 mg per mil to 0.2 mg per mil. And then the protein normalization is 0.2 to 1.2 mg per mil. Pull the chat back up really quick. If you load the same amount of lysate across 25 capillaries, how much variation in total protein do you observe? I am not sure. I would assume that it's pretty close. We do have an internal within 15% CV for all of our experiments, but you could always try it.
Yeah, we have tried it, and there's variation that I did not anticipate, which is why I asked the question. I just want to see. So what I'm hearing is 15% CV is what you would expect, and I think's reasonable. Yes?
Yeah.
Okay, thanks.
Although I think some of the other people may have a little bit more experience with doing that across the board, but that's what I've seen just in general.
Henry, just to jump in, Monica's right, spot on. You should be getting pretty low variability, but if you're doing the protein normalization, you'll want to ensure that you are in fact using fluorescent capillaries that have been QC'd to look for scratches or dust particles that may interfere with that fluorescence and cause some autofluorescence. You'll want to also make sure that you're within a good linear dynamic range for whatever kit you're using. And then also it's best practice to prep that reagent last in order just to keep the best stability of that reagent.
Helpful, thank you. We'll try. I'll get back to you if we're not getting better numbers than I see right now.
Yeah, Phil here. I'm sorry, but if I can jump in with just a couple more insights on that as well. You know, the binding of either the fluorescent or the chemiluminescent reagent is going to be, it's similar. I shouldn't say it's similar, but it's non-stoichiometric. So in other words, like if you were doing a Comasi stain, it's not, if it will bind differentially to different proteins in the mix. And that's part of the reason why you want to do total protein, because all of those are going to get essentially averaged together. So the numbers that we've got here are not absolute numbers. I would think of them more as guidelines, because different types of lysates or different tissue homogenates or serum or plasma are going to label a little bit differently. So it's most appropriate to compare them, basically testing them out for those different lysates. That being said, if you look at our app note, if you're testing different tissues, you're going to get more consistency with a total protein than you would with a single reference housekeeping gene. So I guess the intent of this technology is to be an improvement over a single protein technology detection rather than absolute replacement for BCA or something like that. Hopefully that helps.
Thanks.
There are two more questions that weren't answered.
Yeah. All right. So we've got one. We often see that the protein normalization peaks are similar to our target peaks. How can that be explained? Can you elaborate on that one just a little bit for me?
Hello. Yeah, so whenever we do the protein normalization, we see our target protein peaks. The protein normalization peaks are just like a little bit shifted towards the higher molecular weight, but the same size of the peaks, as if they're repeating the same peaks of our target proteins. And whenever we do the same lysates with a different target protein, sometimes those peaks are repeating those target proteins or don't repeat at all. But the point is that whatever the graph for total protein normalization, it differs on the same lysates between our target antibodies and that we're using. And so my question was that, is that some sort of interaction between those antibodies and protein normalization reagent, or like how can that be explained in general?
I'm not quite sure on that. I might have to look at the file. Does anybody have any insights on that?
So if I may, I'm still not quite understanding the situation. You're seeing on 2 different samples.
Yeah, so with some of our targets, we see the protein normalization peaks similar right next to our target peaks, you know, when you overlay.
Yes, okay. Yes.
And in other targets, you know, we have our target peak and the protein normalization graph doesn't have any of those similar peaks to our target protein. And the problem is that why does it differ on the same samples and the same lysis to begin with? And second, you know, if there is that peak that repeats our target protein, it technically increases the whole area of protein normalization. So it makes that normalization biased, right?
Yes. So if I'm understanding it correctly, is there any chance that you have an overexpressed protein that's being picked up by
Not really. No,
Not
no.
really.
These are just, yeah, no, we do nothing with overexpressed proteins, and you know, and it's and these peaks differ in the same lysates between the targets or target proteins. You know, if that peak was repeated in the same lysate sample on different targets, we would expect that, oh, maybe there is abundance of proteins of that particular weight, right? But it changes between each run.
Okay. I mean, that's a fair question. So like Monica said, we would have to review that data. What state or institution are you at?
Colorado State University.
Okay. I put my email on there.
Yep.
Georgia and we can both help you with that question. That's a really good question. I'm Tuan, by the way.
Nice to meet you, Timothy. Thank you.
So there's a couple of caveats. If you're using protein normalization, there will be variability if you use lower than 0.4 mg per mil. Protein normalization, total protein, they always give that very similar profile of the most abundant proteins that you'll see, like a Kumasi stain. So if you're seeing differences of the same sample, it may be the variability of the protein normalization assay itself, and we have to be above the background. So the cutoff would be 0.4 mg per mil. But please feel free to send me your data and we can talk more offline.
Sounds great. Thank you so much. Thank you.
I've got another question on here. We have observed some zero values for protein normalization when running 0.2 mg per mil lysates on high molecular weight kits as opposed to the 12 to 230 kilodalton plates. Why the difference between the 2 plates? Would you recommend using total protein as opposed to protein normalization for the high molecular weight kits? I think just in general, running your 0.2 mg per mil lysate, I would probably recommend the total protein assay to begin with, since you are at the bottom range of that protein normalization kit. To answer the question about the difference between the high molecular weight kit and the broad range kit, I would say that if your proteins are falling under 66 kilodaltons, like the majority of them, if they're under 66 kilodaltons, that could be why you are seeing, less of that come up in the high molecular weight kits as opposed to the 12 to 230, since the high molecular weight kits is looking from 66 to 440 and the broad range is looking from 12 to 230. See.
Are there any other remaining questions that we haven't answered?
Will changing the protein normalization time improve the protein normalization process at all? I'm assuming you're meaning increasing the time for the labeling. I'm not. You could try it. I assume that maybe at some point it could get to a detrimental effect. But I think potentially extending it, you may be able to see some sort of increase on there. But I think most of it would really have to do with your lysate concentration.
Thank you. I also had a question. In your training materials, you mentioned that one way also to improve protein normalization was increasing separation time. Is that still active? Because I feel like those were outdated files. And is that still valuable? And if it is, can you explain why? Would that help in any way?
Increasing, you mean increasing the separation time in order to increase the protein normalization signal?
Yes, yes.
I'm not too sure about that, actually. If somebody happens to know.
So the separation time, some people would increase it to increase resolution between closely migrating proteins or the higher molecular weight proteins to get it migrate lower. But on our platform, we do not recommend that you go more than 31 minutes of separation time. We don't want to run the 1 kD off the capillaries because that's one of our internal standards that we need for the software to calibrate. But we also see a phenomenon that if you run the separation too long, past 31 minutes, you get the splitting of the high molecular weight peaks. Now, this is done at 375 volt for 31 minutes. Okay, we see that splitting. I have seen people run this at 275 for 45 minutes to increase resolution, but this would have no effect on protein normalization.
I see. Okay, thank you so much.
Any more questions?
I'm sorry, can I ask another question? I'm sorry for bombarding you with so many.
That's okay.
Just to clarify, you know, we're storing that protein normalization reagent in -70, right? And then we reconstitute it with a room temperature reconstitution agent. Is that the correct process? Do we have to put that prepared stock or working solution on ice at all? Like, does the temperature matter or time matter in that case at all?
I've actually put some precautions on the protein normalization reagents. It's been a while since I've done it, but I think both are kept in the freezer and then you take it out at the last minute and then you mix it together. Not sure if the reconstitution is at room temperature.
It was even in one of your training quizzes to keep it in the room temperature, and it's in the pamphlets too. And that's why it was like a little bit confusing. So what we normally do is that we reconstitute that agent, that reagent, original one, right? For stock solution, we put it on ice, you know, for like briefly, and then prepare the working solution. And then as we're ready to put on the plate, we put it on ice too. But, you know, just conceptually thinking, you know, it's room temperature versus -70, whatever that powder is in the original reagent. Like, is that at all, like, what would be the official recommendation? Like, how do you do it in your labs?
So if you notice, I put some precautions and recommendations for protein normalization. I would recommend that you load everything on your plate, leave row B empty. When you're ready to do row B, you take the reagents and then you mix it together and you pipette in those wells and run the plate. You don't want to leave that out at room temperature for so long on the plate.
But you have to spin down the plate for 5 minutes.
Yes, so once you go through that, so leave row B as the last thing you do, then go mix up the reconstitute the lyophilized protein normalization labeling reagent. Dilute it as you need to, right, based on various concentration, and then pipette it into the well of row B and then spin it and then run it immediately.
Okay. Well, that's basically what we're doing.
Yeah, don't minimize the time on at room temperature besides the spinning.
Okay, so the temperature is the main factor that why we should speed up the process, right?
Yeah, just just don't bring it out. That is, I would recommend, just don't take it out until you're ready to run.
Oh yeah,
those reagents.
So after the wash buffer, right, addition, like the last stage before spinning down.
Yes.
Okay, thank you.
Before spinning it down.
Thank you. Oh, sorry, if there's no, can I ask another question?
Yes,
on
please go
unrelated ahead. Topic. Well, not related, but not fully related to this. Is there a particular reason why in Compass software, the normalization to total protein is only done to like one of the capillaries, or that we can change, but it's still one capillary? Like, what if you're doing different samples? Isn't it more logical to normalize it to the total protein within each capillary? Is there a way to make that as an option in general?
So the software won't allow you to do that. It's just a default to reference capillary number 2. And the assumption here is that all your samples should be looking equivalent. So you don't want to put your negative sample in reference capillary 2. If that reference capillary 2 happens to be very low level, I would pick another reference capillary. But the algorithm of the software, it actually works out. However, if you want to normalize every sample to its capillary, you can easily take it out into Excel, just copy and paste the data out and do it manually. Well, not manually, but use Excel.
Yeah, that's exactly what we're doing.
Yeah, but it works actually for the normalization, like, with the algorithm using reference capillary number 2, provided that all the samples are the same and you just use reference capillary number 2.
And you're looking for the one with the highest area of total protein, right?
Not highest, it would be the average area, right?
The average area. Okay.
Like looks all across the board the same. I'm trying to put the PDF into this for the protein normalization, but I'm not able to. But I can email it to you after the fact. And it does say that keep at 18 to 28 degrees. So I think most of the time, if I'm not wrong, we keep it in the 4 degrees. Protein constitution.
It's Jennifer here from tech support. So the reconstitution reagent, it does need to be at room temp because it contains the MSO, so we don't want it to be too viscous. That's why it's kept at room temp.
Okay, so it's 18 to 28 degrees, and the normalization reagent is minus 70 to 80. So that's the reagent that you don't want to keep out at room temperature for a very long time. So that's what you'll leave for the last step.
Yes. So technically, we're doing everything correctly, and that wouldn't create any problems, right, for protein effects. Okay.
And then just make sure your lysate concentration is above the background, which is at least 0.4 mg per ml.
Yeah. Thank you so much.
Okay, you're welcome. Are there any other questions at this time? Okay, well, on behalf of Monica and I, we want to say thank you for attending today's talk. Oh, I'm sorry, there's one more. Yeah. There's a couple more questions. Go ahead.
One more question just popped up. Why is the working concentration of protein normalization different in 66 to 440 plates compared to 12 to 230? I don't think it is. I think it depends on the lysate that you're using. And with the 66 to 440, it's only looking at proteins between 66 and 440, whereas the 12 to 230 looks at proteins between 12 and 230. So you may see differences in your protein normalization quantities between the two different plates, just based on where the majority of your proteins in that lysate fall.